Psychedelic medicine, it's really having a moment, a full-blown renaissance. But for it to truly deliver on all that promise, scientists have to solve this fundamental puzzle that sits right at the heart of how we test any new treatment. I mean, we're seeing incredible results from studies using psilocybin for everything from severe depression to even debilitating cluster headaches. In one major trial, just a single dose combined with therapy dramatically cut down depressive symptoms. The potential here is just, it's undeniable. But there's a catch. And that, right there, that's the core question. When the treatment itself is a profound, life-changing psychological experience, how in the world do you separate the chemical effects of the drug from the incredible power of hope and belief? That is the challenge we're going to unpack right now. So here's how we're going to break it down. First, we'll get into the core research problem. Then we'll look at the unique blinding challenge these studies face. We're going to create a checklist for the perfect placebo, and then rate the top contenders. We'll also touch on microdosing, and finally look at how we can solve this whole psychedelic puzzle. All right, let's dive right into our first section. We need to talk about why a patient's own expectations are so incredibly powerful in medicine. You've probably heard of the placebo effect, right? Well, it isn't imaginary. It is a real, measurable phenomenon. Your belief that a treatment will work can actually trigger real physiological changes in your body. And that's exactly why clinical trials use a placebo, a sham treatment, to figure out what the drug is actually doing versus what our powerful minds are doing. Okay, so now let's see how this whole thing creates a really unique and fascinating problem for psychedelic research. Because, well, the standard methods just don't work. Think about it this way. It's like doing a blind taste test between a glass of water and a super spicy chili. Everyone is going to know which one they got, right? In a typical double-blind trial, neither the patient nor the doctor has any idea who's getting the real drug versus a sugar pill. But with psychedelics, the effects are so obvious, so profound, that the blind is immediately broken. Participants know, therapists know, and this is called functional unblinding, and it throws the entire experiment into question. So you can see why a simple inert placebo, a sugar pill, just isn't going to cut it here. What researchers really need is an active placebo. Now, this is a decoy drug that actually mimics some of the noticeable side effects of the real drug, maybe a change in heart rate or a little bit of anxiety. It just makes it way harder for anyone to guess who got what. And that's the gold standard we're aiming for. So just how bad is this problem? Get this. In a major study on psilocybin for alcohol use disorder, both the participants and the therapists guessed who got the real drug correctly over 90% of the time. I mean, when the blind is that broken, you can't really be sure if the amazing results were from the drug or from the powerful expectation of getting it. Okay, so it's obvious we need a better decoy. And researchers have come up with a kind of scorecard to evaluate the candidates. So let's look at the criteria for the perfect active placebo. Here it is, the 5-point checklist for the perfect decoy. First, it has to mimic both the psychological and the physiological effects. Second, it's got to kick in and wear off around the same time as the real deal. Number 3, it has to be safe. And finally, and this one's crucial, it can't have any therapeutic effect of its own for whatever condition is being studied. Now for the fun part. Let's actually see how different drugs stack up against our checklist. We're going to break down the top contenders based on all the latest research. First up, we've got dextromethorphan, or DXM. You might know it as a common ingredient in cough syrup, but at high doses, it produces its own psychoactive, dissociative effects, that kind of feeling of being detached from your body and your surroundings. And that makes it a pretty interesting potential placebo for long-acting psychedelics like psilocybin. So if we look at the scorecard for DXM, it pretty much ticks all the boxes for psychological and physiological changes. Its timing, a slow onset that lasts for hours, is a fantastic match for oral psilocybin. It's generally safe. The only little wrinkle is that a DXM combination drug is actually approved for depression, but that's with daily dosing, so a single dose is really unlikely to have a therapeutic effect. All in all, a very strong contender. Okay, next up is Salvinorin A. Now, this is a completely different beast. It's an extremely potent but very short-acting dissociative hallucinogen. It comes from the Salvia divinorum plant, and its unique properties make it a potential candidate for super short-acting psychedelics, like DMT. And this really shows you that matching principle in action. Looking at its scorecard, yeah, it produces powerful psychological and physical effects. But check out the duration, a super rapid onset, and the whole thing is over in like 10 to 30 minutes. So it's a terrible match for psilocybin, but it is a perfect match for vaped or intravenous DMT. It's considered safe in controlled settings and probably doesn't have therapeutic effects on its own. Our third candidate is one you probably already know, THC, the main psychoactive compound in cannabis. The way it affects perception and mood, and its timing when you take it orally, make it another option for those long-acting psychedelics. So let's look at the table. Oral THC, it produces psychological and physiological changes, and its slow onset and hours-long duration, that lines up really well with oral psilocybin. It's well tolerated, and a single dose isn't really known to have long-term therapeutic benefits. The biggest potential issue here isn't about the drug itself, it's cultural. For people who are already familiar with cannabis, the effects might just be too easy to recognize, which could break the blind all over again. So far, we've been talking about these full perception-altering doses, but the whole blinding problem looks completely different when you're studying microdosing, which is taking these tiny, supposedly subperceptual doses. See, with microdosing, the effects are meant to be really subtle, maybe a little boost in energy or focus, so the decoy doesn't need to be some powerful hallucinogen. Instead, something like a mild stimulant, caffeine, methylphenidate, or even an antihistamine like diphenhydramine could work really well as an active placebo. The big challenge here, though, is that these substances might actually have their own therapeutic effects for things like depression or cognitive enhancement, and that could really muddy the results. Finally, it's so important to realize that just finding the perfect placebo is only part of the solution. The other part is about designing smarter, more robust studies from the get-go. This really shows that you need a multi-pronged approach. Researchers can improve blinding by recruiting people who've never tried psychedelics before. They can also just not tell participants the exact drugs being tested. They can really emphasize that people's responses to these substances vary wildly. And critically, they can measure what participants are expecting to happen beforehand and use that data in the final analysis. So, to sum it all up, the path forward for psychedelic medicine really relies on a two-part solution. You need carefully chosen active placebos that match the real drug, and you need to combine that with smarter study designs that manage people's expectations. This is really the only way we're going to get the high-quality evidence needed for these treatments to become mainstream. You know, this whole challenge leaves us with a pretty fascinating final thought. In a field where the psychological experience is so deeply tied to the therapeutic outcome, it forces us to ask a bigger question about healing itself. If our biology and our beliefs can both create such powerful, real change, where does the real effect of a medicine truly begin?